Cancer, one of the most lethal diseases of the 21st century. It continues to be a challenge to the medical community. Conventional treatments like chemotherapy and radiation can be effective, but they often come with many side effects. Now, imagine tiny robots, meticulously designed, capable of targeting cancer cells at their very core, the DNA. These nanobots offer a level of precision in treatment that traditional methods can't match, potentially reducing the need for harsh chemotherapy and radiation. This is not a dream, but it is true due to companies like DNA Nanobots. Furthermore, these small robots or nanobots are generally biocompatible, as DNA is a fundamental component of all living organisms, and this compatibility significantly reduces the likelihood of adverse immune reactions, which is a common challenge with many cancer therapies. But the potential of DNA nanobots extends far beyond the treatment of cancer. They could be engineered to detect specific molecules or markers associated with various diseases, leading to earlier and more accurate diagnosis. Today, we talk to Chris Lucas, the Chief Science Officer of DNA Nanobots. With a deep-seated background in technology commercialization and DNA nanotechnology, Chris is not just riding the wave of innovation, he's helping to create it. Join us today on Delta as we dive into exciting and rapidly evolving world of DNA nanobots. All right, welcome, Chris. I'm so happy to have you here today.
Yeah, thank you so much, Ruben. Good evening. And first, thanks so much for the invite and having me on the podcast this evening. Very, very excited to be here. And also congratulations to you for your on your success to date.
Thank you so much. Appreciate it. So Chris, like this DNA nanobots, like what is it? Like how do they work? Can you explain for our listeners, like what is this technology, how it works, where is it used?
Absolutely, Ruben, happy to. So yeah, DNA nanobots are programmable, custom designed nano devices built entirely out of DNA, and they can be tailored for biomedical applications, including targeted therapeutics, as well as nucleic acid biosensing diagnostics. It's a targeted delivery, drug delivery platform technology that is both drug payload and disease agnostic. And so we use a variety of different antibody molecules, aptamers for targeted specificity to tumor-associated antigens. And then we're payload agnostic as well. We can add small molecules, we can add oligonucleotides and peptides onto our device as well for targeted delivery.
Gotcha. So for our listeners who don't have like a background in oncology and the cell basics, can we define like what is the target? What you mean by antigen? What do you define?
Sure.
What do you mean by like you can target specific cells? Like what does that mean?
Right, absolutely. So what I mean by antigen is a specific protein molecule that's expressed on the surface of target cells that we're trying to deliver medicine to. And for example, we're interested in monitoring target molecules that are elevated on the surface of tumor cells that are not expressed on normal healthy cells and tissue. And so, for example, we're targeting a certain molecule that's elevated on AML cells associated with poor prognosis in patients and so forth. And we use antibody molecules that are raised to specifically bind to those target antigens on the surface of target cells. And so that's how we achieve targeted specificity.
Yeah, gotcha. So for those people also, like they don't know how to we treat patient when always, or people when they think about cancer, like they always think about like untreatable disease. Like, well, there are some cancers that are still untreatable, but we have lots of new treatment and new drugs. And what are you trying to do is like we actually what like scientists are trying to do is like cancer cell is very different from the normal cells, and they are different by what they express on their surface. If we find that specific target, that specific clock that we can attack, so we can attack specifically cancer cells, and we can spare the normal cells. So in the back in the days, like 20, 30 years ago, we had only chemotherapy. So chemotherapy, it's a group of drugs that kill every single cell, especially the cells that divide very rapidly, which is like
Yeah.
cancer cells. But now we are becoming more toward like, or we are going toward precision medicine, where we can specifically look at the cancer and say, well, like what type of antigen, what type of protein is there on the cancer, and what type of drug I can give that can kill the cancer cells specifically and not the normal cells. But like beyond
Yeah.
cancer, where do you see the application of this?
For non-cancerous cells?
Yeah.
Yeah, no, certainly. For non-cancerous cells, our platform, not only do we have a program in precision medicine where we deliver targeted chemotherapy to AML cells, but we also have vaccine developmental programs as well, where we can deliver a concentrated payload of antigenic peptides and
Oh,
adjuvant
wow.
molecules, specifically to antigen presenting cells, which ultimately help the vaccine work in the best way it can. And so, but perhaps one of the most things we're excited about, one of the biggest directions that we're beginning to undertake is in the realm of non-viral gene delivery. And so with our platform technology, what we're able to do is essentially use a gene that we're trying to deliver to disease cells with abnormalities in a targetable manner. Not only that, but also add editing molecules that will not only take our gene, our DNA genobot, if you will, directly to the nucleus for incorporation, but we'll also edit out specifically mutated genes that we're trying to swap out with healthy genes to restore normal cell function. And so we're so excited about the non-viral gene delivery as well.
Wow, that's very interesting. So like
Yeah.
beyond delivering a specific drug to a cancer cell, for example, I can find a bone marrow cell and deliver the gene of normal hemoglobin instead of like sickle cell hemoglobin. Is that?
That's exactly right. Yes,
Oh,
absolutely. So
wow.
with our, as you well know, there's I'm sure, Rupen, there are major limitations to viral gene therapy associated with the AAV
Mm-hmm.
vector with respect to expense. Massive expenses are associated with generating the viral vectors. There's packaging issues with the genes, the genes that are be targeted to cells. And I think you can only achieve about 4 to 5 kilobases and kilobase pairs in terms of your length of delivery platform. With our technology, there is virtually no limit to the size that we're able to form a DNA genobot with. We're looking at rare disease and all the way up to dystrophin for rare muscular dystrophies as well.
That is very cool. Talking
Yeah.
about cool, like how did you end up in this cool space? Like, what's the story? Why did you choose to start this?
Sure. So, gosh, it was probably about it's about 10 years ago or so where I had the chance as a biomedical scientist to do a postdoctoral fellowship with a mechanical engineer who was a biomolecular engineer working on a platform technology, DNA origami, nanotechnology in particular. And he came out of a lab in Germany that did a lot of the fundamental studies and really essentially carved out the exact methods for the field in order to how to design these guys with CAD-assisted software and to build them in an effective manner and ultimately to purify them. But he was interested in applications. And so me with the biomedical science background, I came into the lab and really helped him get started the cell culture program, not only looking at signaling molecules with the immune cells with the platform technology, but also leading towards targeted drug delivery devices. And so that was about 10 years ago or so. But we knew that if we ever truly wanted to make an impact and drive the technology out of the university to the clinic and really be involved with the entire process, then we would have to form a startup company. The other option for us was to out-license the technology. Intellectual property was something that we struggled with getting off the ground, but finally we were able to file a series of patent applications related to our targeted drug delivery program, related to our vaccine program. Diagnostic biosensing is another thing that another capability of our platform technology. But we knew that if we truly wanted to do our best to see something through from the whiteboard all the way to the clinic, that the best way to do that would be through founding and launching a biotech startup company.
I love it. I love it. I love it. You touched on things that I think about and I talk about, and this is what the podcast is about every single day. So you are a researcher. You have a background in PhD, and sorry, you have a PhD and you're a researcher, and but you realize that to bring this idea to life, you have to create a startup company.
Mm-hmm.
And this
Absolutely.
is something lots of researchers struggle with. I find like lots of researchers get stuck in writing papers,
Yep.
publishing until one of the papers maybe make it to the guidelines or no, but like there isn't transition to
Yep.
conceive a product.
Yeah, couldn't agree more, Ruben. That's something I'd noticed as a researcher, that, oftentimes, it's grants and papers, as you mentioned, grants and papers, but publish the paper, write the next grant, publish the next paper to get the next grant. But that's an endpoint. And so, maybe a faculty investigator will get to the point where they say, okay, we really want to get this to market. And so, they'll file a patent application. But unless there's a true a business lead, I think within the research program who can co-found the company that truly understands the nuts and bolts of exactly and has the expertise of exactly what's going on and exactly how to optimize something to perform. The proof of concept research is certainly at the academic center, right? But once it gets to the point of applied research, and you have to translate that out of the university, because that's where the true support will come in form of investment dollars, in the form of in the form of prospective clients as well, in the biopharmaceutical industry, who can truly have the revenue and the resources to partner with you and drive something forward. Then ultimately that entity could be either the sub-licensee if it's a collaborative opportunity that you're engaging in. That's what we're doing right now in our DNA Nanobots, our startup company, actively looking for prospective clients to partner with them and to help them make their medicine better, work better in a more targetable manner. And so ultimately could lead to sub-licensees, sub-license agreements, as well as acquisitions, right? And so, in order to truly move something past the proof of concept stage to the applied research stage, in my opinion, it needs to be translated out of the university setting.
Can I ask you, and if you feel not comfortable, it's okay, we can skip this question, but like,
Sure.
how was the transition from academia to the business world, to the startup world?
Yeah. Yeah. So that's been, certainly the most exciting part of this journey, Rupen. Let me tell you, got the itch. I got an itch to do a startup company. I kind of had a feeling, as a postdoctoral researcher, that being a faculty member wasn't for me. I don't know if that were just the stress of trying to get tenure, grant writing all the time, and all the stressors that come with being a junior faculty member or not. But ultimately, what I wanted to do is truly do everything I could do to move a technology from the whiteboard, a proof of concept laboratory setting, onto the applied research and true preclinical development, and get something all the way to investigational new drug filing and first in humans clinical trials. And I thought that the best way to do that would be to serve the biopharmaceutical community. Not only do we have a pipeline of therapeutics that we're currently developing, we have an indication, a targeted drug delivery device for AML, a cancer vaccine for melanoma, and ultimately we're starting to develop a pipeline where we have non-viral targeted gene delivery devices for rare disease. And so it really, I had to do this. I just got an itch for it. That I wanted to see something through, and I've learned in the transition from the academic world to the business world that certainly if you want to be ultimately controlling as much as you can in terms of the developmental pipeline, the strategy, the exact experiments that need to be done, then the startup biotech startup world is the way to go. It is a very challenging world, right? And many challenges that the first time I pitched this idea just to a faculty member, when with a few students that were colleagues of mine and myself, He looked at me and us and said, "You'll be starving in a year with this business model. You guys don't have anything." And what he was getting at is, and looking back, that frustrated us at the time. That was back in 2014. But we didn't have any intellectual property. We didn't have any patent applications that were filed. And so securing intellectual property was a major challenge for us. And it really, we took that feedback, and we knew that if we wanted to get our technology out of the university setting, we had to secure IP. We had to file patent applications, and develop an intellectual property portfolio. And certainly, but the true common thread challenge that we've seen, not only from our tech transfer office, drug development institutes within our university, et cetera, and reviewers, that the hardest part of all of this was convincing people, key opinion leaders, that what we had was something of value. That was and still is really the major challenge that we faced. And in order to get over that challenge, it's so important to develop a team of individuals that not only believe in the potential of the technology that's being developed or the therapeutic that's being developed, but also the team that's behind it. And if I've learned anything throughout this process, I've learned that you really have to build a very strong team in order to convince folks, because otherwise it's going to be very, very difficult. And of course, data speaks for itself as well. So not only a strong team, but also doing the key proof of concept experiments that'll de-risk the technology for investors as well. And
Amazing.
in addition to that, intellectual property team, the next big thing, especially in the business world, to really convince investors that you have something of value, you have to confirm that there is a large unmet market need. That is something that I've learned, and we've been through a series of I-Core Innovation Core programs, not only at the state level I-Core at Ohio, but also the NSF. The NSF has a national program where it's really like an entrepreneurial training system for academics and scientists in the STEM field that ultimately want to become entrepreneurs. Of course, I have not gone to business school. For me, it's been on the job training, but certainly finding that unmet need, unmet market need, is something that's so important, and you have to get out and talk to the market. You have to talk to people. That's the only way. That's one of the big disconnects, I think, in the academic community. We always thought, oh, we have the best XYZ, whatever that is, if therapeutic, or we have the best diagnostic, we have the best platform. But unless you go out into the real world, people who are willing to spend millions upon hundreds of millions of dollars, or $1 billion to develop a drug or more, right? 1.5 to $2 billion, then you have to get that feedback from those people, the people that have the resources, the people who've been in the private sector developing these therapeutics, developing these med technologies, diagnostic, what have you. That's truly one of the things that I've seen that you have to have when you go talk to investors is there a market, and two, do you know who your customers are, right? And so those are two major things that I've learned throughout this process, no doubt.
This episode is brought to you by Mesh AI. Mesh AI is a scheduling software for healthcare workers, including medical students, residents, physicians, and nurses. It alleviates administrative burden by leveraging AI algorithms for scheduling shifts. Mesh AI automates the creation of equitable schedules, efficiently assigns the most suitable staff for each shift, and facilitate easy shift trading among healthcare professionals. Mesh AI is more than just a scheduling tool. It's specifically designed to cater to the unique training requirements of each residency program. By integrating advanced features, it not only manages the schedules, but also supports the specialized training needs of healthcare professionals, ensuring that the training needs of each residency program are met. You touched upon very interesting things, and I saw a common theme: convincing people to be your team member, convincing investors, convincing, and building teams.
Yep.
So let's say I'm an academic person doing my PhD. I have a great idea.
Yep.
I woke up one day, I listened to this podcast.
Right.
What would you do? What would you say to the younger self of you, the younger version of you, how to start building teams and convince them to work with you to achieve your dream?
Absolutely. No, great question, Rupen. I appreciate that. So, certainly the first thing I would do, as a trainee, as a PhD student, as a postdoc, if I have a new idea, a testable idea, have some data to support it and so forth, I would just run to my faculty member, my PI, and say, I believe that this is commercially viable. Do you agree with me? And hopefully the answer is yes. And at that point, the second step would be to engage with your technology and commercialization office. A relationship has to develop over time with a licensing manager within your university's tech transfer office. And so again, that goes to the convincing part. You'll have a series of meetings and you'll do an invention disclosure, and you'll meet with your technology licensing manager multiple times throughout the process, of filing a patent application. But ultimately, in order to get an idea out into the real world, out so an investor can spend hundreds of thousands of dollars or more to get the idea off the ground, you have to have intellectual property protection. And the way to do that is by really just not being afraid to reach out and talk to people. Like talk to your faculty member often about your dreams, talk to your licensing manager. And the next key piece that is certainly in the puzzle for us is, once you get a technology patent application filed, the next key piece is to engage a business lead. So somebody that really has know-how in the biotech space to license technology out of a university setting, right? Somebody that served in as a biotech CEO before, or at least in the C-suite as a chief science officer, et cetera. And that also is a key piece. So it's really building in two things: building a team, and then really educating yourselves in the business biotech world. Even pursuing an MBA, I think, is a great idea as well. I know it's more school and so forth, and for me, it's been on the job training. But certainly, I've seen that there's certain critical pieces that go through the pipeline of getting an idea in the laboratory actually out into the real world. And certainly starts with relationships between your PI, licensing manager, and business lead, because it's your business lead who will serve as your virtual CEO. You don't have any funding, right? So you're essentially a shell company. You don't have any funds, but you can file an LLC in Ohio for $99. I'm sure it's something similar in Michigan where you are, Rupen. But that's just to form a company, right? But to actually launch operations, get pre-seed level funding, and go out and engage investors, you'll need to license the technology out of the university. So the IP positions, the patent applications, the portfolio, and that's your business lead. Your business lead will, be the point person in their relationship with your licensing manager and other key opinion leaders within the technology and commercialization office at the university. So at that point, you'll hopefully get good feedback from your licensing manager. And oftentimes you won't sit in the negotiations because there's a conflict of interest. Likely you still have an appointment within the university. Maybe you'll have plans to go halftime at the university, 51% if you want to write SBIRs, in your company and so forth. Those are small business innovative research grants. But ultimately it's the business lead getting the license. Once the license is in place, you can then approach investors. You're going to look for early stage, high risk, high reward type of individuals. They're usually high net worth individuals. They're called angel investors. A lot of times they fly under the radar. They're maybe not, publicizing what they do and so forth. And usually you'll network in somehow. So it's to the early stage investors. It's really important just to keep having conversations with key people in the university's entrepreneurial ecosystem.
I love it. I love it. This 10 minutes were full of lessons. I love it. This is very, and lots of food for the thought for sure.
Yeah.
Um, I want to understand more about the company.
Oh,
So,
sure. Yeah.
like in the context of like the buzz word around like nanotechnology, we hear this a lot. Like, um, what do you think? Let's say I'm like an investor, or there is an investor listening here. Like,
Mhm.
what does or how does DNA nanobots distinguish itself from others in the field?
Sure. Yeah, a great question there. Gosh, I think it would, in one word, I would say it goes to control. One of our leading benchmark technologies that we're always competing against is the liposomal formulation, right? And so where we stand out, and I think where the true power and the potential of DNA nanobots to deliver medicine to ultimately save lives is one, the unprecedented programmability and control in terms of number and placement of not only targeting molecules to take the delivery system to disease cells and therapeutic payloads, but two, the uniformity of a constructed targeted drug delivery platform critical in the drug development process. When I talk to folks often, every time I'm at a liposomal talk, I'll ask the presenter, if you could estimate, how many drug molecules are in one liposome. And often they give a range. They say it's difficult to quantify. And it's true. It's very true. They're kind of unstructured, not a uniform size, and that ultimately leads to limitations and off-target effects that we don't want, right? Like ultimately in targeted drug delivery, we want a low dose to achieve maximum efficacy with a minimal off-target toxic effect, right? And so that's the ultimate goal. And so we can achieve, in contrast, in the DNA nanobot system, if we want a single targeting molecule on one edge of, say, a rod-shaped structure, we can achieve that. If we want two on one edge of a rod-shaped structure molecules, we can do that. If we can, load them in the center, we can load them on the side, and it's complementary base pairing that allows us to do that. So we have a certain level of control that's unprecedented in terms of molecular functionality that cannot be achieved with liposomal technology. And so that's where I think we distinguish ourselves. You can think of it a lot like Ruben, like Lego pieces. We can piece, a two or three, length Lego piece exactly where we want, on a larger rod shaped structure. We can do the same thing with our DNA nanobot technology. And so, yeah, that's really what I think distinguishes us from other benchmarks is the control. And the same thing with the drug payload. So, for example, we have a tunable drug loading device where we can achieve a 50% loading, and we know that means if our device is a certain base pairs, let's say if it's 7,200 base pairs, it's 50% loaded, then we know that roughly 3,500 or so single drug molecules are within one device on average. And so we can estimate that in a much more accurate fashion compared to our benchmarks, the
I
liposomal
love it.
application. Yeah.
So also again for those people like who don't know what liposomal means, like in
Sure.
simple words, or you can go ahead and explain it. Like
Yeah.
what is liposomal technology? Because I'm
Yeah,
familiar with
yeah,
this. I
absolutely.
use it
Yeah,
every
thank
day for
so
my
yeah,
patients.
liposomal yeah, yeah, for sure. Thank you, Rupert. So yeah, liposomes are really you can almost think of them similar to bubbles. They're a phospholipid bilayer, much like a cell that has a charged residue and a nonpolar or a fatty residue, hydrophobic residue. And what folks do is they use this micelle or a bubble, if you will, to encapsulate a certain amount of small molecule drug payload. And ultimately, what that does is and it's been shown, to work well and so forth. And in the clinic, there are, however, side effects that prevent more widespread use and unpredictable drug release rates is another thing. That's another area that distinguishes us as our drug release rate is very predictable in contrast to liposomal formulations. And so, but certainly small bubbles, they're really difficult to control a uniform size and in a uniform drug payload within each liposome.
Gotcha, gotcha.
So, yeah.
Um, and now like we can't do an any episode podcast without talking about AI, where do you see artificial intelligence fitting with this technology or with DNA nanobots? And how do you think that AI can accelerate the development of this technology?
Sure. Yeah, absolutely. Great question. AI is coming to the forefront of everything, right, in our lives, and in DNA nanomedicine, it's no exception. I would predict that AI could absolutely feed right into our workflow in terms of custom programmable targeted drug delivery devices for the treatment of cancer, right? For an example, if AI can provide us with the most accurate combination based on epidemiological data, treatment outcomes, clinical trial data, what have you. If AI can provide us with the most appropriate combination of, let's say, two or three therapeutics. So I think in terms of AML, so you think of a daunorubicin and cytarabine, let's say, and just two different types of molecules. Not only that, but based on an individual patient profile, if they have some kind of target molecule that's elevated on only cancer cells in that particular patient, then that gives us exactly what we need for a platform drug delivery system. We can design a device with two and combinatorial therapy and all on a targeted device, and we can design, construct, and load our nanobot for not only precision medicine in the context of targeted drug delivery, but for personalized care. And it's AI that would give us that information, Rupen, I think, as quickly as possible. And so, another one of the benefits of our technology is that we can create these DNA nanobots in a matter of seconds. It's just simple Watson-Crick base pairing at C, A to G, C to T, right? And we can do this very quickly. We can decorate the molecules, load the molecules, the A to T, C to G, very very quickly in just a matter of hours. We can decorate appropriately. So
we are talking about devices, and like I think people, when we talk about device, we think about like a huge factory and there's like a production line and they're like, but like this is more on the molecular level.
yeah,
So for someone who never been in the lab, how would you explain? Because like this is not something that everyone knows about it, and I really
sure.
love the technology. I want people to hear more about it. Like,
Yep.
how do you design a DNA device?
Sure. Yeah, great question. So we use a computer-assisted design software program called CAD Nano. And it's also been more user-friendly technology that's came out of Professor Carlos Castro's group, excuse me, called Magic DNA as well. And these are custom design CAD software programs that really allow the DNA origami expert to build these guys. And let's say we want to design something simple like a plate, like a single layer plate. The first step we would do is approximate what that shape would look like in terms of cylinders, where each cylinder is represented by a double helix of DNA. And so how we do the routing then, we use what's called a scaffold. And it's scaffolded DNA origami, meaning we begin with a single stranded loop of DNA and we'll route that scaffold through the entire approximated structure. And so the next thing that we do once that's routed through the entire base of structure, we know the sequence. We use M13, MP18 bacteriophage DNA it's a bacteriophage genome as our backbone, if you will, or a scaffold of the structure. And then we can use that computer-assisted design software to populate the entire entire reverse complement sequence of our starting material, of our scaffold. Once we do that, we make them to an optimal length, which is 30 to 50 bases in length. And then we just simply read out the reverse complement sequence and then order a series of oligonucleotides. And once the oligonucleotides come in, we have our purified circular DNA. We put the two components together. We heat the reaction. We slowly cool the reaction. And then based on long range molecular interaction, simple base pairing, A to T, C to G, we will form a predefined shape structure at the nanoscale.
Gotcha. Gotcha.
Yeah.
Are there any uses currently? Are nanobots commercially used in currently in treating diseases or cancers?
Sure. So to the best of my knowledge, no. We are as a field really starting now to move out of the proof of concept phase of research to the applied research phase. So in terms of therapeutics, we're in preclinical development all at this point. There's been a series of patent applications by our laboratory and others over the last 10 years in particular that's really elevated in a very robust way. And so now our applications are beginning to, you know, go through the applied research phase, go down the regulatory pathways. If it's a diagnostic device, it'll be likely a 510(k) regulatory route, with the FDA, comparison device and so forth. If it's a therapeutic, we'll go down the regulatory pathway that's investigational new drug with the FDA. We'll, satisfy all the submission inquiries and so forth related to an IND and application, but ultimately to move towards a first in humans clinical trial. So to date, no, we're certainly out of the proof of concept phase now in the applied research phase.
Gotcha. You mentioned the FDA, and I always love to talk about them in every episode.
Okay.
I don't know where are you at right now in navigating relationship with the FDA, and what if not, if you haven't started the relationship yet, like
Yep.
what challenges do you anticipate?
Right. Absolutely. Great question. So, related to our diagnostic applications, we've engaged with regulatory experts, I guess folks that engage with the FDA on a pretty regular basis, both at the Ohio State University and in the private sector. So we've not yet engaged with them directly yet. A little bit premature, both with our diagnostic devices and our therapeutic devices as well. But certainly there are folks out there that, it is their expertise, to help, biotech innovators engage with the FDA and really learn exactly what's needed in terms of, a data profile in order to present to the FDA.
Gotcha. Gotcha.
So,
Um, we were talking about the biotech space, and and one of the healthcare is hard, but biotech is much harder. Why do you think it's very hard to innovate in this space?
sure. No, great question. I think about this all the time, especially in our early stages. And again, Rupen, I think it goes back to convincing, the right.
Exactly. It's
It's
scary. Like
convincing,
whenever
convincing,
people hear
convincing.
medical
Yep.
device, okay, but whenever people
Yep.
hear biotech, like, no, let's
Yep.
pause. Let's think about it. It's like,
Yep.
why people get scared when they hear this word?
Sure. Yeah. No, no, I think it's, one thing it's new. I think folks are, very apprehensive at times to, certainly invest, a lot of capital in particular, if you're talking about investors into something new, unless it's been de-risked. And, I think anytime you have a new therapeutic, people are going to want to know first and foremost, of course, is it effective, but is it safe? Right. And so if the answer is no, then or marginally, then you may need to make modifications and so forth and moving forward. But certainly it's so hard to innovate in the space because it's so hard to convince folks. If you're, we were mentioning that there's a grant mechanism called the Small Business Innovative Research Grant mechanism through the NIH as well as the NSF. And it's really difficult to convince reviewers of our applications that what we're proposing is it has merit and whether or not it's certainly going to work, right? And so that's been tricky. And now in terms of investors, they're going to want to know if there is a market need for this technology, especially at the institutional level. And one thing with trying not to go on a rant and so forth, but early stage companies, they really struggle in terms of finding the right investor. And when they approach institutional level funding, like any kind of venture capital firms and so forth, even those that say that they're early stage, you say the word platform and they're going to cringe a little bit. And I think the reason is that they view that as a group that's unfocused. That's one of the key feedbacks that we've gotten from institutional funders is platform not focused. And our response to that is, well, what do we do? We develop targeted drug delivery devices. We do this in the precision medicine space for chemotherapeutic delivery. We do this in the vaccine space for targeting to certain immune cells. And now we're developing a targeted gene therapy approach, right? So targeted delivery is our common thread. So we would say, no, we are focused. And so it's the dialogue that comes with time. And also another thing that's tricky for early stage is unless you're a post-revenue company, unless you're you have clients, you have customers, institutional folks sometimes won't even talk to you, unless they know your background, your story, and so forth. But so yeah, certainly it's the hardest part again is convincing reviewers, investors, especially institutional level investors or other key opinion leaders that you have something of value. Not only does it work, but there's a market, large unmet market need for that. And so now we are starting to change some minds, I think, of investors, one of our angel investors in particular. We have to keep convincing them, hit technical milestones, hit business milestones in order for them to keep helping us and keep supporting our efforts, right? But one of our recent bright spots certainly is our marketing campaign. Our go-to-market strategy is to partner with biopharmaceutical companies and help them create a more effective device, target more targeted capabilities, less off-target side effects. And we use this campaign led by Hypersocial is the name of it. And it's certainly happy to share the website and so forth. But it's a phenomenal tool. It's a LinkedIn-based campaign that allows that allows folks in the biotech startup space to reach out to 800 prospective clients per month. It's an incredible system that they use. You give them certain keywords and so forth, about what you're looking for, the areas of medicine that you're interested in, and what you do and so forth. And then the certain positions of the individuals that you're hoping to contact. If it's a business lead or a business development person, a chief science officer, chief executive officer, et cetera, they will help you have a LinkedIn-based marketing campaign, using AI that ultimately automatic system that reaches out to people. And so, to date, we've reached out to over 1,500 people, and we have a 51% connection rate since mid-November. And so, this marketing is just it's just fantastic because certainly business and I would also say to colleagues out there just getting started, business happens on LinkedIn. There's no doubt about it.
I
That
agree.
is yeah, absolutely. And so a targeted marketing campaign where you're reaching out to lots and lots and lots of individuals, not only through the prospective clients, but also through prospective investors. You can also use a similar approach, which we're starting to navigate as well. So that's been super helpful for us. In a matter of 8 weeks, we have we've talked to probably about 25 companies now, and just even over the holidays and so forth, they've been responsive. It's a great system for marketing, no doubt about it. So
I love it. I love it. And we are talking actually just before the last question. We touched base upon like lots of things, and you mentioned like you applied for grants and and you understand the technology and
yeah.
you know how to write papers, you know how to convince people because like doing research is convincing, is like letting people believe in your idea and your results.
Mhm.
But when we build teams, and this is something we didn't talk about, and maybe we can now talk about it, like
Sure. Yeah.
I think the rationale is always you have to hire your weaknesses. You have to find people like it doesn't make sense for you. You go find another person who knows the technology very well.
Sure.
And you mentioned and you talked about finding someone who understands the business part. Like if
Mhm.
I want to start a company today and I have a background in academia,
Mhm.
who are the team members I should reach out to and make sure they're on my side?
Sure, absolutely. Great question. So yeah, it certainly would depend on the field and so forth, but you want to have, if you're an academic, if you're a principal investigator, let's say, and you want to be involved with a startup company, but you have an academic appointment, it's really, really valuable to have somebody in your lab that has co-developed the technology with you. That somebody that has a technical expertise, is super important. So in our case, we have, chief technology officer. Patrick Halley is his name. He's been a phenomenal partner to me for the last 11 years now, trying to drive our make this dream come true of starting this company. And what Patrick is so good at is not only does he have come with a business background. He's worked in finance. He's worked in all different kinds of sectors and so forth, including culinary arts and financing, and he has a portfolio type of career. But it's really like you said, it's working with people who have complementary skill sets. So one, a technical expert, two, like myself, I'm not too shy when it comes to going out and talking to individuals, right? The convincing part, and it's all about relationships. It's all about getting to know a prospective investor, prospective client, and so forth. And also, you want someone that is able to help you determine quickly, whether or not there's an unmet need in the marketplace. They say fail fast, right? And so having a business lead who has a knowledge base of the market that you're looking to enter with your technology is so important. So not only do you need a strong, technical team and a technology that works, but you also have to have a very driven business lead who not only is exceptionally good at convincing people that the technology in question is of value, but also someone that can engage the marketplace and lead a marketing campaign that allows the technical folks to succeed. And that certainly is the complementary technical and business pieces that have to merge together. And often, what we're seeing is the business folks sometimes tell us in workshops and so forth, the technical people, not to use lingo, not to use jargon. And apologies if certainly I know I have, and I'm glad you've helped me through and break things down to a more clear level. But business folks use their own jargon, and so you have to learn that jargon. And
I agree.
as technical folks, as you're progressing. And so, but certainly, I think really, complementary skill sets. You're exactly right. I think it comes to, you want to hire people that are smarter than you in certain areas, right? And that working together as a team where people are excel, at what they're great at, and then they'll work with their teammates to get feedback and how to address whatever's there. But complementary skill sets in a wonderful harmonious relationship between the technical founders and a business lead is so important to have.
I love it.
So
Last question.
yeah.
Why are you doing this? What drives you?
Oh my goodness. In it to save lives. I mean, plain and simple, to save lives, to improve the quality of life. You know, it's it's been a dream of mine ever since, on my undergrad days when I was looking, what career route to go down, is to fight disease behind the scenes. And if I'm involved on one team that ultimately drives, a therapeutic or a targeted delivery system or what have you, all the way to the clinic to get approval to actually save lives, then I've done my job. And I really think with this career track, I'm able to oversee, at least so far, I've been able to oversee the entire process, right? But in short, it's to save lives. we talk about this every day. It was our effort from, drawing back on the whiteboard back in 2013. We're drawing our little, our Trojan horse idea is what we call our device, our targeted delivery system, to the clinic. And ultimately it's to save lives and improve the quality of life. So no doubt about it.
I love it. I love it. Thank you so much. Thank you so. I really appreciate you being here, and I learned a lot. I learned a lot. Even the technology is still new for me, and I learned
Sure.
a lot from you today.
Awesome, Rupin. Well, yeah, I can't thank you enough. I really appreciate the opportunity, and it was a great discussion, and thank you for your questions. And I think it's great what you're doing, and you're empowering folks out there. And I'm also happy to engage with anyone, at any time. So please, feel free to share contact information, especially from students. I also, I didn't mention, but I also helped co-develop a undergraduate entrepreneurial training program and a wet lab experience for underrepresented minority students. And that has been, something that I'm really passionate about, is helping others, succeed. So
I love it. All the links that lead to you will be down in the comments. So make
great.
sure people, everyone who's listening to this, check the comments, check the descriptions, LinkedIn and website, and all the materials that we talked about. It's going to be in the description below. Awesome.
Excellent.
Thank you so
Thanks,
much.
Ruben. Absolutely.